Thank you all for being here. Thank you, Jordi, for being with us. It's an absolute pleasure to present you Professor Jordi Soriano. I met him a few months ago in a conference, and I was absolutely amazed about the interdisciplinarity of his work. He's developing, in my opinion, a critical work for the next 2 decades, a mixture, very promising mixture of biomedicine, neuroscience, and physics, or if you want, complexity theory. Also, with connections with robotics. So it's a nice mixture for every of us. All of us will find something that is familiar to our work. He studied physics in Barcelona, and he actually developed a PhD in fluid dynamics. So physics, traditional physics. But then he moved soon to biology and biophysics, doing he developed two postdocs, one in Beirut, where he did developmental biology mainly. And then he went 4 years to probably the best place in the world to study and do research on real neuron cultures, the Weizmann, very, very prestigious Weizmann Institute in Israel. He came back to Spain 7 years later with Ramón y Cajal. And 2015, I think, he has a permanent position in the University of Barcelona, where he directs a group on neuroscience. And also, he collaborated to create, and right now he's the deputy director of the University of Barcelona Institute for Complex Systems. Complex Systems, that's it. All right. Also, he's a very bright speaker, so I'm sure that we're going to have a great time now. Please don't hesitate to ask whatever you want. And please, Jordi, thanks a lot.
Animal. So imagine that I take a fragment of tissue from the center of the body column, where we have stem cells, basically. I take out that piece of tissue, I dissociate the cells, then I wash the cells. So I eliminate any kind of signaling. Then I put the cells together, I centrifuge that thing, and I put it there in the lab, and I just wait, okay? I just wait 24 hours and see what happens. And what happens is this: the cells self-organize, they start to oscillate like this, and then in about 24 hours, a new axis is formed, okay? A new axis is formed, and then the animal finally completes regeneration with the formation of the tentacles and the rest of the body. So basically, this animal is amazing because it has an extraordinary regeneration capacity. And people have been investigating that because not only when the multicellular organisms emerge, they started to show complex behavior, but also they had an amazing capacity to regenerate themselves, check for their structure, and that regeneration was gradually lost as we became more and more complex, okay? And this is very nice, but also something fascinating for me in the context of my work is that Hydra has neurons, okay? So even the oldest animals that we can, let's say, see in nature, they started to develop cells-like neurons as soon as possible, yeah? And one of the reasons for that is that you need fast communication between the cells, okay? To that extent along the entire body, and that is fundamental to avoid prey. So you have to hunt and to avoid to be hunted, yeah? And then you need basically to process stimulation and communicate as fast as possible. So basically the fascinating thing is that neurons are fundamental for the development of multicellular organisms to survive. So you need basically stress. Neurons seem associated to the fact that you need stress from the environment and from other animals to keep going and improve in your survival. And then in this context, I recommend you very much this review. This review is fascinating because it explains the evolution of the neurons along the last 1,000 million years. And the message of this review is that the fundamental ingredient to have a neuron are these channels: voltage-gated, sodium, calcium, and potassium channels. So if someone wants to investigate the emergence of complex behavior and then intelligence, one has to look for these voltage-gated ion channels. And my question is any one of you exploring these ideas? Is anyone investigating the possibility to observe this kind of ion channels in possible living organisms somewhere in the galaxy? Is a question that, you know, I make to you, and I think it would be very nice to discuss. Yeah, but one of the beautiful things is that people knows that in using genetic studies, that actually there were very ancient potassium, calcium, and sodium channels, very ancient, about people think that more than 1 million years ago, 1,000 million years ago, and that evolved a long time. And actually, you have this, let's say, symbols here, meaning that there was a breakthrough from a genetic point of view in those ion channels that became much more efficient. Okay, yeah. But in any case, they appeared long time ago. People believe that at the beginning of life, one of the first things that nature did was this kind of ion channels. Okay, so I think for me it's very fascinating. Okay, but in any case, I work with neurons, so now let's go to neurons. And I will explain today the concept of neuronal cultures. So neurons are very important. They appeared a long time ago, and of course they form our brains. And in my case, I study them from the point of view of complex systems. And you have to imagine that you can take neurons from different sources. I will go to that in a moment. And then you can put them on a glass cover slip. And that's why I brought somehow some of my toys here. Okay. And then so you can get an idea of the scale of these cultures. So you put neurons on this one, yeah. You put neurons on this kind of wells, so you can take a look, so you can get a feeling of the real life, the real size of these systems. And then one of the amazing things is that neurons are able to connect to one another, and in just let's say 1-2 days, they start to connect very quickly, and in 5 days they show activity. So actually, this means that once this network of connections between neurons is formed, you get a system that is able to activate itself and show what is called a spontaneous activity. So you have to do nothing. The system by itself will will make a network, and in just 5-6 days, you have a system that is able to activate and maintain itself. And this is for us very impressive, because this is one of the most beautiful examples of a system that it's able to self-organize and show complex behavior by itself. Okay, and it's beautiful because it's very small. As you see, you can put it in a relatively small cavity, and you can play a lot with what makes it very fascinating for us. Yeah. So to continue, I have to introduce very briefly some concepts of neurons and how they work. By the way, if you have any question, please stop me. Okay, ask me anything. Perfect. Yeah. Yes. Okay.
You said it.
Perfect. Thank you.
I'm going to do it now because then you're going to jump onto different things. But I am very curious about why potassium channels appeared first. Is there any, I don't know if this is known, but is there any preference for potassium to form a protein channel before?
Oh, they are somehow the easiest.
Okay,
they are somehow the
simplest.
Easiest. It's the simplest, yeah. The simplest voltage-gated ions channel that you can produce is based with potassium.
Okay,
yeah.
okay. Thank you.
Actually, you can look at the review, but the fascinating thing about these ion channels is that they were not created, let's say, or nature did not make them for communication. They were made to regulate the ionic concentration inside the cells initially. Yeah,
okay.
But then they evolved. They were so useful that they evolved to use them for signaling. Yeah.
Okay. Thank you very much.
Thank you. Okay, good. So then I have to introduce very basic concepts just in case. One is that when one thinks about neural networks, one has to think about three ingredients. One is the intrinsic dynamics of the neuron. This means that neurons basically, which are represented here, neuron receives inputs through the dendrites. They integrate this information, and when the information passes a threshold, then an action potential is emitted. Okay, so conceptually, the neuron is on the resting state. which is shown down there. Then the neuron gets excitatory inputs that increase the memory potential, or inhibitory ones that decrease it. And when the total reaches a threshold, the neuron fires and goes back to the resting state. This is the fundamental dynamics of the neuron. And it's important to be aware that you need excitatory and inhibitory neurons because they play the role of traffic lights in the sense that if everything activates together, the system cannot convey information. If neurons just fire randomly, there is no information there. So you need a precise wiring and control of information to be able to send something meaningful. Yeah. So you need excitation and inhibition. The neuron integrates information and passes it. And then something else that is very funny somehow is that when the electrical signal reaches the end of the presynaptic neuron, it is converted into chemical signaling that goes to the postsynaptic cell. And this chemical signaling is mediated through vesicles. Yeah. And these vesicles actually are released by accident a lot of times. Yeah. There are so many because the vesicles are continuously produced. They are stored in the presynaptic cleft, but they are also released by accident. And that creates a natural noise in the system. So that's why this explains why you have a neuronal culture, and the neuronal culture is able to activate all the time by itself. Yeah. And finally, something important is the network. Of course, you need a network architecture that is, in principle, thought to do a particular function. Okay. And this is what we call the complex network. So essentially, you have the intrinsic dynamics, the noise drive activity, and then the network tells you or gives you the capacity to process information and do something interesting for the animal, in this case, to survive. Okay. So depending on how you wire the network, you get one activity or another. Okay. Good. So these are the essentials, but the important thing is, okay, that's very cool. So how do I get data? So there are two ways to get data. One is to use this kind of systems. This is called multi-electrode arrays. I will give it to you in a moment. And this system, where you put the neurons in the center, this is like a CMOS chip. And this CMOS chip has, in this case, 4,000 electrodes. So you record simultaneously what happens in 4,000 electrodes. The funny thing, or the great thing, is that you can interface this system through electronics with something else. In this case, and that's why I brought this kind of friend here, that if you want, you can interface your neuronal culture with a robot. Okay. And we'll play with that later. But in principle, the point is that you can record activity in this nice system. And if you want, yes, thank you. And if you want, you can actuate on that system. So this is one measure, one approach. Another one is what I showed you before, these kind of flashes of the system. And these flashes correspond to the use of what is called genetically encoded calcium indicators. Sorry, I wanted to play it again. Yeah, no, okay. So people use what are called genetically encoded calcium indicators. Conceptually, what they do is to infect the neurons with a virus that encodes for a fluorescence protein that allows me to see activity in the system. So basically, I have a system that is electronic and a system that is optical to record activity in my cells. And then I can decide, depending on the interest and the technology that I need, I can decide one or the other. Okay, good. So for the case of the system with electronic communication, this is the kind of behavior that one can record in the laboratory, where each of these yellow flashes correspond to the activity that we see in the culture. And then something beautiful that we are doing. Sorry, sure, yeah. No, the strong yellow spots correspond to neurons activating. Yeah. So and then the blue violet background is noise.
That's noise. Okay.
It's noise, exactly. Yes. And then so one of the thank you.
It's like synchronize everything there, like they're always firing at the same time.
It's a good question. It depends on the wiring that you have of the neurons. I will go to that in a moment. Yeah. But yeah, it's true. You can get synchronous dynamics or something that is much richer. Yeah. And then something beautiful that we can do with this system is, for instance, to couple it not only with robots, but what is called neuromorphic chips. So neuromorphic chips are essentially a new emerging technology in which what we do is to develop hardware architecture, okay, that is designed specifically to simulate neurons in your system. Okay, so basically you design like a GPU or a similar system, but the transistors are designed in such a way that you have like neuron-like systems in your chip. So easily in the market you can buy these chips, and they have about 1,000 neurons, the simplest ones. And IBM developed some that have 2 million neurons. 2 million neurons is the equivalent of the brain of a bee, for instance. And the fascinating thing for us is that you can communicate neurons, real biological neurons growing in this system, with artificial neurons growing in this electronic device. Okay, and the nice thing of that is that you are developing hybrid interfaces or hybrid systems where biological systems and electronic ones are communicating to one another, and in principle they're able to synchronize, to change information, and these things. Okay, so it's something that, at least for a physicist, is very, very fascinating. Okay, and good. So then, with all these things in context, some questions that emerge that are interesting are, well, can we build biological networks whose dynamics approach the one of the brain? Of course, we all will have this brain inside of us, okay? And we know that the brain works very well. It's capable of many things. But of course, if I put the neurons in the class covered sleep that you saw before, the system can be extremely boring, okay, and can do nothing interesting. So can I build neural networks in culture that resemble the brain? Yeah, that is one question. Then, are these neurons resilient to damage? Can I kill some neurons and see whether the system survives or not? And finally, are these cultures a good system, a good model for medicine and artificial intelligence? And these are some of the questions that my lab is developing. And basically, we combine different ideas, that is physics of complex systems, medicine, and artificial intelligence, and reservoir computing, and finally, neuroengineering. I will go to that briefly, but basically, these are the tools that I work in my lab. So in case that you are interested in neuronal cultures, I recommend you to look at actually a review that I wrote one year ago. Yeah. In this review, I explore basically all the things that one can do with neuronal cultures. So conceptually, starting with our neurons in the laboratory, we can investigate spontaneous activity in the sense that we know that the system is able to activate by itself. So we want to understand why that happens, how stable is this activity. Then resilience to damage, we can kill neurons with a laser and see what happens. We can simulate everything in a computer, which is called computational neuroscience, to try to mimic the experimental behavior and understand it. Then we can develop ideas in complex networks to model the structure that we have. We can do neuroengineering, where what we do is to try to put neurons in places that we like, okay? So we have circuits that are more interesting and brain-like. And finally, we can develop therapies to try to cure diseases, okay? And actually, to get an impression of how interesting all these explorations can be, each of these numbers here represent one of the publications in our group related to these aspects, okay? Okay, good. Then when one has in mind the idea of building neuronal cultures that are as similar as possible to the brain, the first question is why? Why do you do that? Well, the reason is that, as the colleague asked in a moment, that if I prepare neurons in culture, as you can see here, the typical behavior now the typical behavior is this one, okay? So the system, so when I connect neurons in a random way, a neuron basically sees other neighbors and connect with them. Basically, I always get this kind of synchronous, highly synchronous behavior that is far away from a normal, let's say, activation. And that actually would correspond to what we would call an epileptic attack, an epileptic seizure, in the sense that all neurons suddenly are very synchronous with one another, okay? But this is somehow pathological. If you look at the brain operates in a way that is something like this. Sorry. It's so difficult to find the mouse. I want to put the video because it's nice. Okay, so this is now finally. Okay, this is important that you see this video because you can compare the dynamics here that is very rich. So different areas activate locally, and sometimes the entire system activates globally. And this is much richer than what you have over there. That is totally boring. Okay, on off all the time. So there is an obsession to try to build networks that resemble the ones that we have in the actual brain. And the reason for that is that if I want to develop technology to develop actual treatments, actual cures for diseases, I need a model whose dynamics approach as best as I can the actual dynamics of the brain. And one of the reasons is that imagine that I want to study epilepsy, right, in culture. So if the system is by itself epileptic, I have a problem. Yeah. So I have to escape from that. And another reason is that our brain works in a fascinating way that is called integration segregation balance. And the idea of that is in this toy brain that I have here, each color represents areas that carry out particular functions. Okay. So for instance, here we have the visual cortex. The visual cortex is in the back. The auditory cortex is here. On top, we have the sensory areas that process information from a stimuli. So what the brain does is to process information independently in each of these circuits and then integrates all the information together to make sense of the reality. So the system is all the time switching between segregated state and integrated one, all the time. It's switching all the time between the two. Yeah. And this is very nice because if we achieve that in culture, then we are approaching a system that dynamically is very similar to the brain. And that is very helpful because if something goes wrong, because due to a disease with this network, maybe it becomes more integrated or more segregated. And we can investigate that and try to find a cure. Okay. So this is why we need desperately to try to make networks that approach as best as we can this integration segregation balance. Okay, good. So with this in mind, conceptually, we can do neuroengineering strategies to try to design building blocks that allow the neurons to connect in richer ways. And then now finally, I explain where do we get the neurons from. There are actually two possible sources. One is called rat primary cultures, and rat primary cultures are very cute because they are very cheap. Okay, that is not, let's say that is not an obsession, to save money, but it's just that when you prepare neuronal cultures, when you prepare neuronal cultures, primary cultures mean that you basically take a dead rat, to extract the neurons from the dead rat, and then if and very quickly you dissociate the neurons and then put them on the glass cover slip. And the funny thing is that in 5 days typically, you get activity in the system and you can record and get data. This is very nice. And for 100 euros, you can get about 50 cultures. It's relatively cheap. And then the problem is that you're using animals. People don't like that. So I'm one, and actually even dead animals, but there is a tendency now to eliminate animal use. So in this context, Professor Yamanaka in 2012 developed a technology that is called induced pluripotent stem cells. And in this case, human-induced pluripotent stem cells, in which you can take cells from your skin, then you can make those cells pluripotent, and then you can differentiate them in whatever you like. In this case, neurons. So this is a technology that is really, really amazing. And actually, you can prepare in vitro cells from different people. Okay, and that is fascinating because that allows you to study particular genetic diseases. So if I put in vitro a genetic disease and then I have cells from a healthy person, I can compare how both networks evolved, and I can try to develop cures for the one that is diseased and see whether it approaches or not the healthy one. Yeah. So this is amazing technology. The problem is that it's very, very expensive. It's super expensive, and you need about between 1,000 and 5,000 euros to prepare 50 cultures, more or less. It's a factor 10 or 20 compared to the primary cells. Yeah, but this is the technology that people wants to use, and actually is the future in neuroscience. Okay, good. And then in the context of neuroengineering, I was saying before that we want to build some kind of building blocks that destroy this kind of homogeneous, boring behavior that we have here. Okay, that is due to the fact that neurons basically connect to their neighbors. Okay, yeah. So and we basically, what we do is we put obstacles or either squares or tracks or even three-dimensional neuronal cultures. And that allows us to break this isotropy and makes the system much richer. And actually, I wanted to share with you some of the toys that we have in this direction, because you can touch it actually. So basically what we did, we used very simple technology, printed board technology, to put neurons, put this, and then I will show you. For you, another one. Here I have more. Yeah. And then actually what I'm giving to you is, then we can pass it, you can pass it to the other people. What I'm giving to you is different plastic masks, okay, that have different shapes. Well, I will pass it, thank you. That they have these plastic masks, have different shapes. And then the nice thing of them is that you can grow neurons on the surface of these plastic masks. So neurons basically have to follow very intricate path to connect to one another. So in this way, in a relatively simple approach, you can build networks that are far away from this homogeneous distribution. Yeah. And the amazing thing is that you can actually make systems whose dynamics is much richer. Yeah. So conceptually, you have to imagine that as a physicist, I'm always looking for simplest scenarios or simplest scenarios that give me interesting information. Good. And then just to finish this part, one has to get data, and to get data, one simply look at the neurons in the camera, for instance. Yeah, one has the neuron in the camera. From the camera, one gets the fluorescence traces, and by analyzing these peaks, one can get the activation of the neuron here. And this is basically the data that we use to later on get information on the dynamics of the system. So the same thing, the fluorescence traces. From the fluorescence traces, we get the activity in the network. So this is the neuron ID as a function of time. The yellow areas represent groups of neurons that tend to activate together. And finally, if you have the necessary technology or analysis tools, you can, for instance, build a network of which neurons are communicating with which ones. So in this case, the dark lines correspond to neurons that are strongly exchanging information to one another. Yeah. And then, of course, the beautiful thing of this analysis in the context of complex networks and physics is that you can, for instance, compare a healthy and a disease culture and see from a statistical point of view, yeah, to see which are the differences that they have. Any questions so far? No? Okay, good. So then let me just show you a repertoire of possible cultures that we can prepare in our laboratory. So the top one is the one that is very synchronous. Then if we put neurons together forming a small aggregates, then you see we have an activity that is very different, in which different units activate either alone or in small communities. Okay, and this is very nice because this resembles somehow the brain in the sense that you have areas that process information and then exchange that information with the rest of the system. Yeah, but the important thing, I insist, is the fact that you can completely disrupt Yeah, thank you. So the point is that, yeah, you can completely break this on-off synchronous behavior and have something that is more closer to the brain, okay? And then, of course, if you go a step forward, instead of putting neurons in these kind of spheres that are shown here, instead of putting neurons in these kind of spheres, you can also put them in designed areas like the ones here. So you can design small squares with neurons inside. And this system, is much more accurate in the sense that you can really tell which neurons connect to one another. And for instance, you can define thin corridors around here. Okay, you can define thin corridors that allow you to connect the different modules with precision. Okay, so in this case, you have here on the bottom left, we have a system that is self-organized, And here the system that we engineer, okay, to have a precise connectivity. And finally, we can make a system in which neurons connect along tracks, for instance. So you really break the isotropy of the system. And then the system is, as you can see, very, very rich, very similar to what we see in the brain in the context of dynamics. So the point is that by using different ideas of neuroengineering, we can jump from a system that is conceptually very simple, in which all neurons activate in a synchronous way, yeah, on the top left, to a system that has a much richer repertoire of dynamics. Okay, questions so far? Yeah. Thank you. Some can be isolated, but the majority of them are connected to other neighboring aggregates. Yeah, but it depends. In this case, we design it to make it totally self-organized. So we just put some drops in the cover glass, some drops where neurons will start attaching, and then they form bigger, smaller aggregates, but they normally connect to the neighbors. Yeah. Some question? Yeah. Exactly. Correct. What happens in the brain when exactly, but you combine it with integration. Exactly. No, it's a dynamic thing. It's a dynamic. Yeah, remember that inhibition is always there. You have to think that you have excitation and inhibition, yeah, and the brain is able to activate inhibition in some particular areas to isolate them from the rest. So conceptually is that the brain and this culture actually are dynamical in the sense that you can control excitation and inhibition to regulate the connectivity and achieve meaningful information processing. Yeah. Okay, good. So then, so this is the kind of repertoire that we have. And then probably you heard in the news the concept of organoids. Do you know what an organoid is? Okay, but just very quickly, it's an emerging technology in the sense that you can put these stem cells I was talking about before, you can put them in 3D spheres, and in some cases, depending on the technology, you can replicate the structure of brain areas. So basically, conceptually, you are building mini brains in your laboratory. Yeah, and actually, this is interesting for us thank you. This is interesting for us in the context of medicine because now there is a big effort for blind people to restore vision by building retina from scratch using this technology. Yeah, so you put cells, then you differentiate the cells with the correct layers that the retina has. Okay, with all the different kinds of cells, and in principle, because you can extract, remember, you can extract this IPS. Okay, these pluripotent stem cells from a particular person. You can generate a system that replicates retina and will not have any kind of problems when you insert it to the person because the cells belong to the same person. You follow? Yeah. So this technology is amazing and is one of the big promises for medicine in the next years. But also the amazing thing and also scary thing is that you can build what is called mini brains. Yeah. And then the debate today in the community is, okay, you have mini brains. So wait, are those human-like things in culture or they are not human? So what is the limit? If you make an organoid very big, is that like a brain, like a real brain or not? So there is a lot of debate on that. But in any case, the fascinating thing is that people is developing technology to demonstrate that these mini brains are able to compute information, classify numbers, for instance, do some tasks that are similar to an actual computation. Yeah. Okay, good. So, okay, so then just a very quick thing, and is that maybe some of you touch this kind of fractal structure. Okay, so the thing is that this is like a physicist dream somehow that, okay, let's combine different ideas. So I want to maximize, let's say, the activity, the richness in the activity repertoire of my neuronal cultures. So I grow them in this kind of fractal patterns with the goal to achieve the maximum variability in areas with neurons that connect to other areas. Yeah. So I have maximum variability because one area connects with a small one, intermediate areas connect with either big ones or small ones, and so on. And this is very nice because in this case, we can use then statistical physics and see whether these patterns and the dynamics that you have there of the neurons approach the dynamics of the brain. Yeah. So basically, I don't know if you work. Oh no, sorry. Yeah, I will put this just to show you an example. So this is the culture. This is the culture as reference. Okay, the homogeneous one, very on-off, and there is the one where we put this fractal pattern, and you can see sometimes the entire system fires, sometimes only one region. You will see that sometimes only the central square. At some moment, central square, come on. And now, well, no, now was the opposite. And now, okay, yeah. So at some moment it will appear, the central square. But the point is that the system is somehow we discovered that is maximally rich. And I don't know if you are familiar with the world of criticality. The criticality concept is that the brain does not operate functioning in a random way or all neurons activating together, but something in the middle that is called the critical point that is shown here. Yeah. So this region, the subcritical, will be random activity. The supercritical will be that everybody basically synchronizes, and the critical means this point here means that you have the maximum repertoire possible of activity in your system. This is something that in the context of statistical physics is a very powerful, a very fascinating topic, and we try to explore that with our cultures. And basically, as you can imagine, we are looking for power loss. Okay, this is the characteristic signature of criticality in a system, and I will just go very quickly on that. But conceptually, what I wanted to show here is that it depends a lot on the method that you use. But basically, you look for power loss, and conceptually, if in this case the exponent is 2, everything is trivial. If the exponent is 1, means that the system activates in a random way. So basically, this exponent 2 means that the system is fully synchronous, this fully random, and something in between means that the system is rich. And in this case, for our fractal cultures, we are in this region with alpha around 1.7. So the system is rich. And then what we did was to compare with data of actual neurons, in this case in the mouse. And then we observed that, as you can see here, the exponents were very similar. Yeah. So conceptually, you see that the blue line corresponds to the actual brain of the mouse or the rat. So actually, the good thing is that by preparing these fractal cultures, we can achieve a dynamic behavior that is very close to the one that we have in the brain. So conceptually, we are getting neuronal cultures that are very good as models. Yeah. And then what do we do with these models is something I want to stress in the last minutes. Yeah. And one of the things that we can do and that are very fascinating is to first observe if the system is resilient or not to damage. What we do is to use this very scary microscope. Yeah. This microscope is a multimodal microscope that allows us to both visualize activity and also use a laser to kill neurons there. So with this system, what we do is that we basically target one of the groups of neurons, one of these aggregates, and see if the next system is able to recover from this damage. And just let me show you what happens. So this is before damage. You see the system is activating everywhere. You see activity everywhere, basically, yeah? And then when I kill this aggregate here, basically you see the entire area is silent except this small corner over there, yeah? Basically, everything is silent except the corner that you have on the bottom right, okay? So then we waited few minutes and we observed whether the system was able to recover or not. So basically, we had the system here. This is our original network before the damage. Everything is super connected. Everybody's talking to everybody very strongly. Then we kill this aggregate here. This area gets silent, and slowly, you see, in few minutes, everything reconnects again, except the dead region. Yeah, this means that neuronal cultures are, in general, extremely resilient to any perturbations, in this case, physical damage. And actually, this is nice because we developed this technology actually to help medical people to develop therapies, treatments for people that has an ictus. Yeah. And ictus corresponds to the loss of blood flow in a particular region of the brain. And then we use this data as a model, and we explain to the doctors that, well, first the system will regenerate, but if you develop additional ideas like chemical action or electrical stimulation, it will recover faster. Oh, yeah? So in this direction, we are producing very simple models that are helping the medical community to develop better strategies for treatment. Okay. And then the second example I wanted to put is this one. I will skip this one. I wanted to skip this one. And the second example is that we can use IPS for actually exploring what happens in some diseases. So conceptually, the idea is that, because I told you before that we can access different cell lines, we can access the cell line from a person that is healthy, that is shown here. This is a healthy donor. Yeah. And then we prepare the culture. This is our control. And then we can go and get data from people that has Parkinson's disease with a particular mutation. And then this is our PD culture. And then what we can do is use CRISPR technology to genetically edit the cell and have the isogenic PD cell, PD line that in principle is cured from the disease. Yeah. And then we can compare the three cultures and see whether the PD and the iso PD are similar in behavior. This means that we were able to cure the disease, yeah, and the impact that it had on the dynamics. And actually, we got an award of the City of Barcelona for all this work in the last years. So let me show you very quickly what happens. So this is our control. You see that we have a lot of activity, but let's say a strong synchronous activity is not very, very abundant. Okay, I can just use this technique to quantify how many neurons activated together in a time window, and you see most of the time is below some kind of threshold. Okay, so conceptually, the system is activating. You have collective activity, but it's not very strong. And if I go to the control for the PD, yeah, if I go to the case for the PD, you see activity here is much more synchronous, meaning that the system is pathologically talking too much to one another. Okay, this is like epileptic behavior. There is too much synchronization. And then when we go to the isogenic, the cured cell line, you see we are able to restore somehow the dynamics that we had in the control scenario. And here we don't have anymore these super strong activations. And if they appear, they're very, very rare. Okay. So conceptually, we can also use our technology, our exploration with neuronal cultures to find cures for diseases and help the medical community to advance in some treatments. Yeah. Some questions about this part? No? Okay. Then to conclude, yeah, I wanted just to very briefly comment on a couple of aspects on artificial intelligence. As I told you before, we are developing technology in which we grow neurons in these multi-electrode arrays that are, well, somewhere. Okay, yeah. And then the important thing is that we can combine them. We can combine that with technology like the organoids, retina development, and see whether we can make a communication between electronic circuits and biological ones. You have to imagine that if you want, sorry, it's here. You have to imagine that if you want to build, or if you want to help a person that is blind, you cannot just put a camera, a CCD camera, and then send that information to the brain. That will not work, because the way that a camera encodes information is not the same as the brain, okay? So you have to do something, yeah? And this something is maybe to couple first the information from your camera to a neuronal culture. So the neuronal culture will process that information, and then you can send that biological-like signal to the brain to make it understandable, yeah? Some colleagues in Japan are actually developing in this technology, and they manage to help blind people to really see with the help of a camera, yeah, interfaced with an electronic device. So it's very cool. And of course, you can also develop implants in the sense that the organoids that I developed before, you can prepare the organoids and then implant them on the person. And in principle, with sufficient stimulation, yeah, if you manage to induce plasticity in that system, yeah, then the new retina will be able to process information correctly and send it to the brain. Yeah. So there is a lot of technologies in this direction that my lab is contributing to try to help people. Yeah. So, and then of course, another fascinating thing is that artificial intelligence, is very expensive. You need huge. Actually, now ChatGPT is approaching actually the amount of neurons in our brain. So it's growing very, very fast. Yeah, but you have on the order of 100 billion neurons. This is really a lot. And actually, you need the energy of a city. Well, what's the city? Terrassa is a city in Barcelona, but I don't know. After Madrid, which other city you have in the surroundings? About 100,000 million people, or 100,000 people, sorry. Okay, so Móstoles could be an example. Okay, thank you. So imagine that you need the energy that Móstoles is using every day for using ChatGPT or training ChatGPT. So it's an absurd amount of money. And actually, and you know that there are many companies that are currently building new ChatGPTs almost every week, right? So people is getting crazy in the sense that there is a huge amount of energy that is needed just to feed these systems, right? So people in the European Commission launch projects to try to see whether we can do the same things more or less, okay? The same things that ChatGPT is doing with biological neurons, yeah? So imagine that you have a biological neuron, let's say 10 million neurons in culture, and can you train them to do, for instance, image recognition or some kind of artistic thing and save energy from ChatGPT and company? At least you can launch the idea and try to explore it, okay? And this is one of the projects I have in my laboratory, in which basically we want to prepare cultures and try to teach them to do something or recognize numbers, for instance. And this is what we do, and that's why I also brought my robot, because one of the things that we are doing is that if we are able to teach a system to do something, in principle, we could couple neurons growing here with the robot. So the robot has sensors. In this case, the sensors are very simple. Well, this robot is very simple, but it costs just 50 euro, and you can play a lot with it. In this case, we put a sensor, we put these whiskers, so it looks like a rat, but we can also put, let's say, cameras. We can also put touch and many other things. Okay, but conceptually, when I turn it on, basically what happens is that if I touch the whisker, the system changes in direction. Okay. That is what happens. It's programmed like that, but conceptually, what we want to do with our cultures is that we can technically send the information of the whisker to the neuronal culture, and the neuronal culture can actuate on the wheels. So in principle, the good thing is that we are trying to see whether the system is able to navigate in an environment just using the whiskers. Actually, there is a question. You are first. Yeah, sorry. Yeah.
Also, like train the system with you program the control first, and then you do the training of the neurons with both the inputs and the outputs.
Yes, but it's very difficult. The training is extremely difficult. Okay. And I will show you a video. Well, actually, I have a question, but actually the mystery, how do you do that? I'm sorry, training is the most important, let's say, secret for the people that is working on these ideas, because there is no a single path to train the neural culture to achieve the goal of learning navigation, for instance. It's extremely difficult. So I have a question. That's a question. Okay, but let me just put you an example of this, because it's very cute. Okay, so first maybe I think that maybe you read this article in the news that people in Australia managed to connect the neural culture to the Pong game. The Pong game is I will show in a moment. And they manage the culture to learn how to play Pong. And this is the game. Have to go to the second 45. So basically what you do here is that you prepare your neural culture like in a chip like this, and then you stimulate. You provide stimulation, electrical stimulation. So basically what you are doing is that you induce plasticity in the neural culture, right? And then you keep stimulating in different areas until the system starts to behave as you like. It's not so obvious, but it works. And then 45, more or less here. And this is the culture playing the Pong game. And it's quite okay. Yeah. It's not easy. And then, of course, the secret, and it's a big fight with these people, is to understand exactly what they do to make the culture able to play the game. And if you tell with them in private, they will tell you that they don't really know. They try different approaches. Okay, sometimes it works, sometimes it doesn't work. So it's extremely, extremely difficult. But in this direction, I will also put a second video, and this is the end of the talk, in which I put a robot. This is done by some colleagues in Japan, in where they have, you can see here the robot with the culture on top when it starts to walk. Now, you see the culture is on top. So in this case, the robot has a camera, and the goal of the robot is to try to find the destination, which is the thing that is emitting light. Yeah. And the point is that the more and more you play with the neuronal culture and the robot together, the better the robot is performing. So it looks very clumsy, but after a few days of training, it very, very nicely avoids the obstacles and finds the path to destination. So it's fascinating, but also very scary at the same time. Because if the robot is able to do that, it's like an animal then, or not? Has some rights or not? Yeah? Okay.
So it's learning.
Huh?
It's learning.
It's learning. Yes, yes, yes. But it's very nice. The robot is performing very, very well. Yeah. And I wanted to stop here just with this video. Let me just thank the people in my lab that is working in these aspects. I will skip this. I will skip these things, but just want to thank my people, okay? And that is working every day to make all these experiments real. Okay, so thank you very much for your time.
Whoa, amazing talk. Let's just have some questions.
Thank you for the talk. It's really amazing. I have got a question about synchronicity, and you say it's pathological, and that's usually true in the grown-up brain. But we know, at least in mice, and I guess that it also in humans, when the brain is developing, there are phases in which there's synchronous activity, which is I don't know if widespread to the whole brain, but quite extensive. This all guesswork, but do you think that it's possible that the synchronicity might be a phase of your culture's developing structure of its own?
Yeah, I mean, synchronization happens. Yeah. Synchronization is important, actually, for broad communication across different brain areas. Yeah, so during learning, you also have synchronization in different systems. It's true. So, but I mean, the normal operation of the brain is not synchronous behavior, but it just occurs when it's needed to exchange information globally. Yeah, but it's true. You're right. The synchronization is one of the phases that the system undergoes when learning or exchanging information. Yeah, because for you, synchronization is particularly important for your no. Yeah. Yeah, but it depends. But some circuits, when they develop and mature, they are actually switching between local activity and global one in the context of synchronization. Both aspects are necessary. For full synchronization by itself all the time is considered pathologic. Yeah. Thank you. Thank you.
Thank you, Jordi. It's amazing and a little bit scary in this technology.
Scary, yeah.
Well, but it's okay. I understand they have many possibilities for the future for sure. But I wanted to go back a little bit to the principles of this, because neurons are cells and
yes,
highly evolved cells, even in this simple animal. The point is, I don't know, perhaps it's a nonsense that it has already been done, but I don't know if it's worth to try to make to build your own neurons. I mean, can you make vesicles that you can implement with these ion channels, calcium, whatever channels you want, or whatever? The point is to start to build your neuron from the beginning.
Oh, wow. That's a very good.
Make any sense of this?
No, it makes sense. I think it makes sense. People that is trying to build artificial cells from scratch, they are also developing these ideas of which are the basic ingredients that you need for an artificial cell to communicate with your neighbor. You need chemical signaling for sure, but also you need this, in this case, this voltage-gated signaling, because it's much faster than chemical in principle. But yeah, people is playing with that.
Yeah, but you can somehow design your own channels, because again, this potassium channels, or sodium channel, whatever, are current proteins. So they have a lot of evolution behind them. So the point is, you can even invent your own protein.
That's a good question. That's why I was proposing, or I was launching the question, because it's true. You could maybe find alternative to that, right? So in other planets, things could develop, and instead of using these gated ion channels, you could use some other protein, right, or some other construction. It's true, but I don't know. I think when people is investigating alternative to life, people have this in mind. But the problem of the point of my message is that you need, if you want to have complex life, apparently you need something that allows you for very fast communication, and in principle, this voltage gated ion channel to provide that. This was the idea. But if you find an alternative, biologically, that would be great. Yeah. Yeah, thank you.
Any more questions here? Yeah.
Well, thank you for your talk. Very interesting. My question is, when you were plotting the correlation, the power law, what's the math behind that? How do you calculate correlations in that special system?
That I will explain you later if you want. In this case, we use what is called the normalization group. Okay, it's called the normalization group approach. So basically, if two neurons fire together, you change that two by one group. Yeah. So basically, you keep renormalizing the system, and if all neurons fire together, you get a trivial result. If there is a random activity, you also get a trivial result. But if you have maximally rich repertoire of activity patterns, then you get a power law. But in this case, it's renormalization group, and you can use other approaches like avalanche dynamics, count how many groups of neurons activated together, and so on. So there are different approaches. Yeah.
Okay.
Yeah.
And second question.
Yes. Yes.
You were in your cultures, you have neurons, but I guess you have other glial.
Oh, yes. I simplified things a lot.
So you have more biological stuff, or just neurons? Yes,
correct. I simplified things to make it to try to make it clearer. But yes, you have glia cells, astrocytes, that those are specialized cells that what they do is to help neurons to work correctly, and also exchange, chemical signaling. They provide, the feeding, let's say, to the neurons. In general, they nurse the neurons, but also they help in keeping the entire network together. Yeah. So glia is one of them. Correct.
But none of them are involved in that synchronization or that.
Yeah.
Or yes.
Actually, yeah. This is a good point, because for instance, many diseases that people thought that were caused by the death of neurons are actually caused by malfunctioning astrocytes, that because they don't work correctly, they kill somehow the neurons that they should take care of. Yeah. So what happens is that, for instance, if astrocytes don't work correctly, then the neurons tend towards an excessive synchronization. That you are correct. Yes. For instance. Yeah.
Thank you.
Thank you.
So any more questions here? If not, we have one last on Zoom. I'll read it. And
okay, good.
So this question is from Andres, and he says, very interesting talk. Would like to ask, related to your last slides about IE, I wonder about the similarity between the architectures of biological neural networks and artificial neural networks used to train IE models. So do you think that we will have to change the architecture of the artificial neural networks to mimic the biological ones? Or it will be the opposite, that is, modifying the biological neurons to mimic the artificial perceptions?
Oh, that's a good question. Thank you. No, actually, when you think about artificial neural networks like the ones from ChatGPT, basically what you do is that you start with a gigantic random network, and then you start disconnecting some connections or creating new ones or reinforcing new ones, yeah, to create an architecture that is able to compute information. Yeah, but this is the basic idea. So yes, you have to modify the original network to make one that is able to compute information in the way that you like. And in the context of biology, what people is trying to do is to investigate from scratch which architecture you can put by default, right? Like in ChatGPT or similar systems to accelerate this kind of learning for a particular task. So for instance, if you remember, I put a system with different communities, with different modules. So people believe that modularity favors the processing of information. So instead of starting with a random network, let's put a modular network from the beginning. Okay, so this is basically the answer of the question. And it goes in both ways, okay? From artificial intelligence to biology and the other way around. Yeah.
Okay, if there are no
yeah.
more questions.
Jacobo.
Just a comment. Thank you very much for the talk, Jordi. It was amazing.
Thank you.
Just a comment on when you Andres was asking and you were mentioning. Actually, many times in the development of engineering, we have only been able to find the way to do things when we stopped copying nature. For example, when we were able to do I don't know, flying, for example, spent like 200 years trying to understand how birds fly, and then we realized that we had to do something else, something different. So perhaps here should be the same. I mean, I'm not so sure that artificial intelligence should try to copy nature or the other way around. What do you think of that?
Yeah, well, people like nature in the sense that our brain with the same neurons as ChatGPT performs much better. This is somehow the concept, right? So biological computation is in principle much more efficient than artificial one. Well, first in terms of energy cost, and also by the fact that we are able to remember things and keep them somewhere and recover them when necessary. The problem of ChatGPT and company is that when you teach the system to do something, forgets completely about other aspects. So you have to teach it again to do something else. It's called catastrophic forgetting in these kind of systems. But biological systems are able somehow to store information in a much more efficient way and very quickly learn things, forget them partially, and then recover them and learn them again very quickly. So people like this behavior of biological systems to adapt extremely well. And also something very fascinating for us, and is that you saw before that biological systems have this spontaneous activity all the time. They are all the time exploring. But ChatGPT and company, they do not have that. So people is trying to investigate why biological systems for millions of years have been wasting energy to have spontaneous activity going on all the time and not just wire and wait for something to happen. So this is something fascinating. And that's why in the context of artificial intelligence, we're trying to investigate how spontaneous activity can help maybe to learn faster. We don't know yet. But there are many questions that are fascinating. And if nature does that and works very well, will be for something. This is our point of view. Yeah. Okay. Thank you.
No more questions? So thank you so much, Jordi, for coming. And it was an absolute pleasure to have you here. And it's amazing.
Thank you. Thank you very much.
Thank you so much.
Thank you.